fundraiser activities where we work with the Miami Dolphins, the Miami Hurricanes, and the Miami Heat. And if you guys participate in these fundraisers, I can almost assure you that this trip would be extremely affordable. So basically free of charge. You could go on a trip to Honduras for less than maybe $400 for 10 days if you go to almost every fundraiser. So this is an opportunity for you guys to gain over 200 hours of clinical experience, over 100 hours of volunteering, and get a travel. And you could do all this for almost free of charge, or at least at a very affordable rate, depending on how committed you are to the fundraiser. So I'm going to leave the flyers over here. We also have our first general body meeting on Thursday at 6:45 p.m. in GN 101. Again, it's on the flyers. So if you're interested after class, you can come. I'm going to leave them over here. And, yep, hope to see many of you guys on Thursday. Thank you, guys.
Okay, I think you have one quick question.
You say that's before the summer courses.
Yeah, it's before. It does not interfere at all with school. So it will be right before summer A starts.
Okay, thank you. Okay, let's go ahead and get started. The first is, actually it seems there's at least one student who actually reads the book, reads my notes, and found a mistake in my notes. It's never happened before. I guess it has to happen eventually. I don't believe that. So anyway, just a small correction. We just went very quickly through the eukaryotic DNA polymerases. And I believe what it says incorrectly is that delta synthesizes both the leading and the lagging strand. This is an example of that kind of bureaucracy of eukaryotic cells. They need two enzymes to do what E. coli does with one enzyme. Okay. So what we want to do today is we're going to continue into chapter 13. We're getting in the area of transcription, probably of the major areas of DNA metabolism that we're looking at: replication, repair, transcription. This is where we'll see the biggest difference in how the system works in bacterial cells and in eukaryotic cells. So there's going to be large areas of overlap, but then there's going to be very specific differences. Shouldn't be surprising, again, since eukaryotic cells have transcription occurring in the nucleus, which is sequestered off from the rest of the cell, versus the sort of open cellular architecture of bacterial cells. So let's start with our usual questions, and then we'll get into the chapter. So from the last lecture, let's try this one. Should be pretty easy, 10 points. We'll just go to 1 minute. Looks like the answers are coming in pretty fast. All right. So the answer is obviously what? Oh, here we go again. Okay, all right. And then from today's lecture, again, we'll go to 1 minute. Okay. And what's the answer? All right. So far, RNA polymerase. All right. We'll know more about that. Okay. So we're in this chapter on transcription. We started last time, and we ended up at this rather, we were at least close to this rather confusing figure. So let's start here and review this because there's some nomenclature here that and some sort of conceptual, organizational aspects of this you have to keep in mind as we move forward. So the key points here are we have the two strands of DNA. Either strand can be transcribed, but for any given gene, except there would be a very rare exception, the transcription is going to be off of one strand, and we have to be able to identify that strand. So you use this terminology of the so-called template strand, or sometimes called the nonsense strand or the Miden strand. This is the actual strand of DNA that the RNA polymerase is going to read base by base and insert the complementary ribonucleotide. Since RNA synthesis is always 5' to 3', just like it is for DNA, then that template strand will be 3' to 5' going in the same direction as the direction of synthesis. The opposite strand is what we call the sense strand. The way I think about it is it's the strand that makes sense. So we do a lot of work for bioinformatics. We download sequences for these genes and such. And we get the DNA sequence, and what makes the most sense to us is the strand that's exactly the same, that is, we use the term collinear, it's exactly the same as the transcript or the RNA, except that you substitute the U and the T. So that sense strand is going to run 5' to 3' in parallel with the direction of RNA synthesis. And so now we can define both this sense and nonsense strand. Okay? Any questions? That's just, we did that last time. Right. In other words, the sense strand is going to have the same polarity as RNA synthesis. They're both going to read 5' to 3', and if you compare this sequence to this sequence and just substitute the U and the T, you get the same sequence. We use the term collinear. Okay. This is just to show you that we have several genes along this piece of DNA. They're not all transcribed from the same strand. So in the shorthand, one is, I would cross out this minus and plus over here because I think that's confusing for the purposes of this slide. We've got three genes, gene A, B, and C. This little arrow is a pretty standard convention that shows you where transcription is going to begin, where the first ribonucleotide is going to begin, and the direction of synthesis. So if gene A is being transcribed left to right, then which one has to be the sense strand? The upper or the lower? It has to be the upper strand because it's also going 5' to 3' in the same direction as synthesis. Gene C is also transcribed in the same direction. So the upper strand is also the sense strand for that. But B is being transcribed right to left. So now which one is the sense strand? The lower one. Again, it's going to have to go 5' to 3'. The sense strand will always be running with the same polarity as the RNA synthesis. Okay, any questions about that? That's where we talked about that last time. Okay. So as we've done so many times before, we have to redefine or more precisely define what we mean by a gene. And a common way of looking at this is a so-called transcription unit. So what we're looking at here are all the DNA sequences that are going to be required to both regulate the expression of this gene and actually provide the template for the transcription. So it's going to have three critical regions to it. The first is a promoter. This is where regulation is going to occur. This is where the whole RNA polymerase complex has to recognize that this is where transcription is going to begin. It's going to also determine which DNA strand gets transcribed. And it also determines where transcription actually starts, where that very first ribonucleotide is going to be added. Usually, these promoters are immediately adjacent to the DNA that they're going to transcribe. So it kind of makes sense. The whole RNA polymerase complex is going to come in, interact with this promoter. We'll talk about that in a few minutes. And it sets the stage for transcription to begin. Here's an important point. The promoter is not transcribed. The DNA sequences that are part of the promoter that we're going to talk about are not represented as RNA sequences in the transcript. Then you're going to have the actual RNA coding region. That's going to be the DNA sequence. Polymerase comes along, adds all the nucleotides so that you get the actual RNA molecule. And then you have to have a terminator. So in contrast to DNA synthesis, only limited portions of the DNA are going to be transcribed, and somewhere there has to be some molecular signals that tell the RNA polymerase, stop at this point, release the RNA, and then begin the process again. So kind of in a cartoon form, this is what you have. Same kind of diagram we had before. This would be like gene A and C from that previous diagram. This lighter color region is the promoter. So it's adjacent to where transcription is going to start. So right here is where transcription begins. So that aligns with the actual transcript. And then transcription is going to be left to right as this is drawn. So this is the whole RNA coding sequence. Then here's going to be the DNA sequences that are the molecular signals to terminate transcription. All right. Now, the promoter is not, as I said, the promoter is not part of the transcript, because transcription is actually going to begin right here. The terminator is usually present in the transcript because the actual ending of the RNA synthesis usually occurs after the polymerase has gone through those sequences. Okay, so that's what they're showing you here. So these are the terminator sequences, so they're part of the transcript. The promoter is not. Okay, make sense? Somebody just nod. Okay. It's going to get worse. Wait till we get to UKARYOTIC. I told you, I'm just report the news. I didn't make this stuff up. All right. So, the transcriptional start site. The start site is the position where the RNA polymerase is going to add the first nucleotide. Okay. Does this same thing happen with DNA synthesis? Is there a start site where the first deoxynucleotide is added? No. Remember, DNA synthesis has these primers. That's going to define the point. With RNA, I'm making the point with RNA polymerases, they do not require the primer. So it can put that first nucleotide in place, and we use the term de novo. It can synthesize RNA de novo. It does not use a primer. So if you look on the sense strand, the nucleotide on the sense strand that corresponds to the first ribonucleotide that's present in the RNA is designated as +1. That's the transcriptional start site. So if we just don't want to make you nauseated, if we go back here, this transcript, let's see. Let's assuming it begin. This isn't really the beginning, but if this U was the very first nucleotide at the ribonucleotide at the 5' end of the RNA, then this T would be the +1 transcriptional start site. So it precisely defines where that first ribonucleotide goes. All right, we were up to here. All right, then we use this kind of jargon upstream and downstream. All the nucleotides that are flowing downstream is the direction that the RNA polymerase is going. So beginning at +1, we actually will number the nucleotides to give them coordinates. They'll be positively numbered as you go downstream and negatively numbered as you go upstream. Upstream will go into the promoter. So it looks like just a basic number line, except there's no zero. Okay. All right. So that's what I'm trying to show you here is this base that's right at this junction between these two colors. That's where transcription starts. That's the transcriptional start site. That's what we call +1. These are minus going right to left and positive going left to right. Okay. There's no zero. Okay. Why don't you let that sink in? So, which of the following is not correct about the process of transcription? Okay. Remember, from this point, you can talk to each other. It's just kind of freakishly quiet today. Because I'm still drinking my Crystal Light with caffeine. Although I haven't graduated to the coffee that's that hyper caffeinated coffee. You know, some brand of coffee, it's got like twice the normal caffeine level. When I was at North Carolina, they had a little coffee shop nearby and had all these different coffees, and one was this Hawaiian coffee. It was simply called Kick-Ass. That was the name of the coffee. Okay. After my little journey down memory lane. Okay, so what's the answer? All right. We got one right finally. Okay. Okay, this part's going to be familiar. The main comments here on the process of transcription, it's very similar to DNA synthesis. So you're going to use the ribonucleotides instead of the deoxyribonucleotides. So we use this R, but more frequently there's just no designation. So if it just says NTP, ATP, GTP, CTP, those are the ribonucleotides. They're added to that 3' hydroxyl on the nascent RNA. So just like we did with DNA synthesis, you cleave the two phosphates, that phosphoanhydride bond to generate the energy to drive the reaction. In that first phosphate, the alpha phosphate is part of the phosphodester bond, and synthesis goes in the 5' to 3' direction. So overall, RNA synthesis biochemically, very, very similar to DNA synthesis. Okay. So what we'll do is we'll look at the bacterial system first, where there's one RNA polymerase. Then we'll look at the eukaryotic system, where in the typical kind of government bureaucracy, they have to use 3 polymerases to do what bacteria do with 1. Okay. So bacteria, we're going to be using E. coli as our example, has 1 RNA polymerase, with the caveat that that primase that makes the oligonucleotide, the primers for the leading and the lagging strands during DNA synthesis is technically an RNA polymerase. But that's the only thing it makes. The RNA polymerase we're going to talk about now makes all of the other, as far as we know, all of the other RNAs in the bacterial cell. These are large, complex proteins, so when you see the term multimeric, it means there's multiple subunits here. So each synthesis translation makes a single polypeptide, but for many enzyme complexes, multiple polypeptides have to then coordinate together to form this multimeric or multi-subunit protein. In bacterial RNA polymerase, the core enzyme, what we're going to call the core enzyme, what the complex that actually can synthesize the RNA, has 2 copies of a subunit we'll call alpha, one copy of beta, one copy of beta prime, and one copy of omega. which really isn't essential for activity, but it helps stabilize the complex. All right. So what do we got here? 2, 4, 5 polypeptides have to come together to form the core enzyme. This core enzyme can carry out transcription. If you put this into an in vitro system, give it all the right components that it needs, a template and such, it can transcribe the RNA. What it can't do is find the right place to start. So it has the RNA polymerase activity, it just doesn't know where the promoter is. And that's the job of sigma, what we talked about in that first one. The question is the sigma factor. So when you see the term factor, you're talking about a protein. All right. What has to happen is the RNA polymerase complex has to know exactly where to interact with the DNA to begin transcription at the right place. This is the job of sigma. So sigma got its name for specificity, so they use the Greek letter for S. If you add sigma to the core enzyme, you create the holoenzyme. Now, the important point here is sigma is only required to lock down the polymerase in the right place. Then it has to dissociate. And then you're left with the core enzyme that will actually do the RNA synthesis. If you had a mutation in sigma where it binds too tightly to, or doesn't dissociate, it'll actually block transcription. It causes the complex to bind so tightly to the DNA that it actually can't begin transcription. So it's just a parking attendant, and then it has to leave. So here's what you have. So here we have the core enzyme, we had sigma, and we get the full functional RNA polymerase, which not only can synthesize RNA, but can also recognize a legitimate promoter and initiate transcription in the right place. Now, to give a little bit of differentiation between the genes, in other words, a major part of gene expression is controlling what genes get turned on at what time and in what place. It's pretty basic in bacteria. What they have is a suite available to them is a suite of different sigma factors. Those different sigma factors recognize different promoters. So in E. coli, you have sigma 28, sigma 32, 54, and 70. They get this designation based on their molecular mass. So, you know, proteins are measured in kilodaltons. So that's what it's based on. The key here is that each sigma factor recognizes a different class of promoters. So for instance, we talked about the heat shock response, but that was in Drosophila. It's a universal response. So if you heat stress E. coli, it normally grows at 37 degrees, you raise it to 42 degrees, you begin expressing heat shock proteins. And that's because the cell makes sigma 32, which now interacts with the core enzyme and recognizes the promoters that are specific for the heat shock response genes. Right. So that's what gives the cell some level of regulation in responding to different environmental conditions. Okay. There you go. Okay. Yeah, so this might be more in lines of an exam type question. So we'll go to 1 minute. Looks like. I'm sorry, what? Okay, we'll go to 1 minute and 15. Okay. All right, so what's the best guess? Okay. Now, I'm sorry, why wouldn't what? Okay. Why isn't it B? Could it be B? Maybe. What would have to happen? All right. The most likely outcome would be C, because the vast majority of mutations are recessive. They lead to non-functional. They are loss of function. You could have a mutation in RNA polymerase. You could have a mutation in sigma where it fails to dissociate, which would be a gain of functionality. It could happen. It would be much, much rarer. The most likely outcome would be C. Okay. So bacteria make it easy. You got the one RNA polymerase with the note that the primase is also an RNA polymerase. In eukaryotes, when they started purifying the RNA polymerases, they expected to find RNA polymerase based on what they knew from E. coli. But in fact, they isolated 3 distinct RNA polymerase activities. They were feeling very imaginative, so they called them RNA polymerase 1, 2, and 3. And the question is, what do they do? So applying the idea of Occam's razor, which I think goes back to about the 14th century, and that is, to paraphrase it, go with the simplest interpretation. So the simplest interpretation, which turned out to be pretty correct, is that each of these RNA polymerases would transcribe a different major class of RNA. So RNA polymerase 1 transcribes and makes the ribosomal RNAs. The one we're going to talk about a lot and we're most interested in is RNA polymerase 2, which transcribes, what I say here is the pre-mRNA. So if you know about, everybody knows about introns and exons, right? Well, you got to get rid of the introns, and we're going to go through this whole splicing and processing step. So that initial transcript, which has the introns and the exons before all that splicing happens, is often just simply called the pre-mRNA. And then you got some of these small nuclear RNAs that we talked about before in the nucleolus, and some of these small nuclear ones that are part of the SNRPs that give you this spliceosome and such. So that's what RNA polymerase 2 does. We're going to mainly focus on it as being the RNA polymerase responsible for ultimately the messenger RNAs, which are going to be the protein coding molecules we're going to be interested in. RNA polymerase 3, you could say in general, makes small RNAs. It transcribes the tRNAs. They're only about 75 nucleotides. The small ribosomal RNAs, so there are actually 4 different ribosomal RNAs. So more accurately, RNA polymerase 1 makes some of the larger ones. A few of the smaller ones are made by RNA polymerase 3 just to make it complicated. And they also transcribe some of the snRNAs. In general, we can just think about it. RNA polymerase 1 does the ribosomal RNAs. RNA polymerase 2 does the pre-mRNAs, which ultimately give rise to the messenger RNA. And RNA polymerase 3 transcribes the tRNAs and some other very small molecules in the cell. These are getting to be rather large complexes. So these are, again, multimeric enzymes. But whereas we had 6 polypeptides for the bacterial RNA polymerase, there's more than 12 subunits in here. And make sure you know all 12 of those. I'm just kidding. Wow, they just like breathe. No, the biochemist will probably make you memorize all 12 of them. Okay. So if we look at this operationally, how does the process of transcription occur? We can break it down into 3 steps: initiation, elongation, and termination, which is sort of your career, okay? But your career works, if you're lucky. You may have no elongation phase to your career before you go straight to termination. Okay. All right. First important point. Initiation is the rate limiting step for this whole process. So a major part of transcription is gene regulation. And a big piece of that is how efficiently is transcription occurring? That is, how many copies of messenger RNA are coming from this particular transcription unit per unit of time? Initiation is the rate limiting step. So the more efficiently that works, the more messenger RNA you're going to get. The less efficiently, the slower. In bacterial cells, this process of initiation occurs, the efficiency occurs over a range of almost 1,000-fold. So we're going to have the initiation step. We'll talk about that in some detail. Elongation we'll go through very quickly. And termination is, you get this whole complex. You've got this RNA that's connected to the RNA polymerase complex. It's interacting with the template DNA. The DNA is unwound. All of that has to come apart, be resolved, so it can start over again. And that's really the process of termination. Okay. So this initiation, as I said, it's the rate limiting step for RNA synthesis. And the whole process is driven by getting that RNA polymerase complex to interact at the proper site on the DNA. That is, that's what the promoter is for. The promoter is that landing strip for the RNA polymerase complex. And kind of a follow-up to what I was just saying, the rate, again, this is the rate limiting step. So what's really changing is by changing DNA sequences, you change the affinity. In other words, how efficiently does this promoter recruit all this RNA polymerase complex? The more efficiently it does it, the faster the rate of transcription. Okay. So if we kind of dissect out one of these typical bacterial promoters, and again, we'll use E. coli as our example. Here's what the promoter is really going to do. It's got to tell the polymerase where transcription is going to begin. It's got to tell it which of the two DNA strands, in other words, which direction to go. Well, so basically 2 and 3 are tied together. Since the polymerase can only go 5' to 3', that promoter has to tell the polymerase you're either going to the right or you're going to the left, and then that will control which strand is actually getting transcribed. So all that information has to be built into the DNA sequence. So as I said, the promoters are immediately, and especially in bacteria, immediately adjacent to where transcription will begin. So it makes sense. You're going to bring the RNA polymerase to exactly the place where synthesis should start. Well, if you start to dissect out and look at the DNA sequence of a large number of promoters, let's say you took 100 the promoters from 100 different genes in E. coli, start lining them up, start looking at them. What you're looking for are what we call consensus sequences. Are there highly conserved sequences that are common to all of those promoters? Here's an important point. Consensus sequences, they're not exact. The consensus sequence is the best fit if we look at a large number of sequences. First, are there sequences that are fairly close to being similar? And second, just how similar are they? All right. So a little bit of the shorthand that you find in these consensus sequences, if you find a particular 2 nucleotides occur at equal frequencies, they'll usually be indicated by a slash. It could be like C/G, something like that. If you usually find a pyrimidine in that position, they'll use a Y. If you find a purine, it'll be an R. And then indicates that there is no base that's really more common than any other base. So you get something like this, where you have this 5' and then this sequence, and this would be a consensus sequence. Think about it in terms of it's the best fit. So here's an example. If we take these actual sequences from bacterial promoters, line them up, and for instance, we see here that we have 3 T's. So T is most common, A is most common. Here, you're equally likely, but most of the time you're going to get a pyrimidine, then you're going to get an A, then a purine. Here, you get what you get, an A, and then C and G occur with about equal frequency. So that's going to be our benchmark, and then all the promoters get compared to that. Yes?
For the Y, why wouldn't it be T/3 instead of just Y?
Because they're all pyrimidines. And same way over here, you got 2 A's and 2 G's, but they're all purines. And you're only looking at 4. Typically, I mean, now with the sequencing, you could take all 6,000 of them and line them up. And you find the consensus sequence doesn't really change much. What came out of that studies, those kind of studies, were a couple of key conserved sequences, and they're designated by their position. So one is the -10 consensus sequence. Simply means it's a sequence that's centered about 10 nucleotides upstream of where transcription will begin. It was originally called the Pribnow box, named after David Pribnow, who discovered it. It's probably still pretty commonly called that, or the -10 consensus. It means it's the same thing. So the sequence is TAT, AAT, and this is, again, if you looked at all of those promoters from all those E. coli genes, centered around that -10 portion, this is what you would see. Also, it's not going to be a perfect match. It's going to be very, very close to that. Yes?
If we are only talking about E. coli.
In this case, yeah. Some other bacteria they'll have the same, it's the same idea, their consensus sequence. For most of the bacteria, for these sequences, they're so well conserved, they're pretty much the same in almost all the bacteria. There's another sequence a little further upstream at the -35 sequence, this TTGACA. Okay, so if you find a consensus sequence, does that prove that they're important? No, it just suggests that they're important. But the follow-up studies, but it tells you, wait a minute, here's my best place to look. So if I start to mutate those sequences, does it in fact affect the promoter? And the answer is yes. So there were genetic studies followed up by looking at mutations in these -10 and -35 sequences, and they clearly affect transcription. And they give you what were called up and down mutations. Most of the time, if you mutate the promoter sequence of your particular gene of interest, and that mutation makes it it does a poorer job of matching the consensus sequence. So in other words, it's moving away from the consensus sequence. These were called down mutations. And in nearly all cases, that mutation resulted in a slower rate of transcription. So things were slowed down. And that's usually what you got. But sometimes you could get mutations that were up mutations. They actually made the gene transcribe more efficiently at a higher rate. And when you looked at those mutations, they actually were a better fit to the consensus sequence. So basically, mutations that give you a better fit to these consensus sequences make more efficient transcription. Mutations that take you away from the consensus sequence give you less efficient transcription. Okay? All right. So mechanistically, what's going on here? This brings us back to the sigma factor. So as we said, the holoenzyme is that core enzyme plus sigma. Not surprisingly, sigma interacts with the -10 and the -35 sequences. That's what sigma is looking at, and it's positioning the RNA polymerase complex based on the location of those -10 and -35 sequences. Now, I'll show you in a diagram in a few minutes. The other is, this RNA polymerase complex is large. It actually covers almost 70 nucleotides. It actually extends out over the transcriptional start site. So don't think about this. This isn't some small little protein complex that's just interacting with 2 or 3 nucleotides. It's a fairly large structure. And what happens is the binding of that holoenzyme to the promoter actually begins the unwinding of the DNA. That's really the rate limiting step. Is before transcription can begin, the hydrogen bonds have to break, and that double-stranded DNA has to open so that the RNA polymerase complex has access to the bases. That is the slowest step in the whole process. I don't know if it's on the slide yet. It's called this transition from the closed promoter to the open promoter structure. And how efficiently it does that is really determined by these promoter sequences. Okay. All right. Just to throw an extra level of complexity on this, the -10, the -35 are the most common, but some promoters have another sequence called further upstream. So as I said before, I think I mentioned before, if you see the term element, and we'll see this over and over again as we go through different parts of the remaining material, you're really talking about a DNA sequence. So some of these have these upstream sequences at the -40 to -60. So that RNA polymerase complex extended out to about the -50. So the part of the polymerase complex can actually interact with these sequences. They're not always going to be there, but if they are there, they tend to enhance transcription significantly. Okay. Here, give you a chance to breathe. Let's try this. So we have a mutation in a promoter of an E. coli gene that makes the -10 region a closer match to the consensus. What would you expect? Yeah. I'll go to a minute 15 on this one. Okay, so what's the answer? So this would be an up mutation, and that it's a closer fit to the consensus, and the effect would be most likely it would actually improve the ability of the RNA polymerase complex to interact with the promoter. Because it makes it a, what did I say? It makes it a closer match to the consensus. As you move away from the consensus, then it would be a down mutation and it's less efficient. As you make it, in fact, basically, the consensus sequence that's worked out is about the strongest promoter you can make. Okay. All right. All right, so this process of initiating RNA synthesis, as we said before, you have to determine the orientation of the spacing, sets up the direction of transcription, locates where the +1 is. And you can see that this is important. It's not only the -10 and the -35 sequences, the actual sequences themselves, but it's their position. So it's kind of like a molecular ruler. Locking down at the -10 sequence puts the RNA polymerase activity right on the proper nucleotide to initiate transcription. If you delete some of the spaces and you move the -10 sequence, keep it exactly the same, but move it to maybe -5. What happens to transcription? Yes. So the transcription will actually initiate a few bases downstream because now it's moved the RNA polymerase to the wrong position. So it's both the sequences and the position of these consensus sequences is critical. So again, no primer required. As we said, okay, so you cleave off the 2 of the phosphates, that gives you the energy. And just like we did with DNA synthesis, that 5' phosphate on the ribonucleotide will now join to the 3' hydroxyl on the end of the RNA. And as I said before, it's now important after those first couple of nucleotides that sigma dissociates or will actually impede the actual synthesis of the RNA because it's locking down the complex too tightly. So sigma factor dissociates, and the RNA polymerase has to unwind the DNA complex, so we have to unwind it. Very similar to with DNA synthesis, but the difference is during DNA synthesis, because replication is semi-conservative, you're permanently unwinding the strands. This is just transient over a very limited area. So after transcription is done, the helix is going to reform it. The double-stranded structure will reform. Okay, but we have the same kind of problems that we had before. You put these positive supercoils in, just like we did with the replication bubble. It's the same process. So you also have to have the topoisomerases. So they have to help control this supercoiling that's happening ahead of the unwinding of the RNA polymerase complex. So to kind of show how this works, so here's the transcriptional start site, here's the promoter, here's the core RNA polymerase, sigma factor. They come together with sigma factor's assistance. The holoenzyme is now locked down in the right place. And so this is what's showing you that quite a bit of the DNA molecules actually interacting with this large complex. And it extends past the point of RNA transcription where it starts, because what it's doing is putting the RNA polymerase activity as part of this complex at this position, and that's how it knows where to put the first ribonucleotide. All right. Then the strands begin to unwind, and you're going to have, and so you have the promoter, it's going to unwind. At some point in here, you're going to add in the nucleotide to get everything started. Sigma has to come off at that point, and then we have the process of transcription, which is unwinding the helix and just sequentially adding the RNAs, which would be the process of elongation. So initiation, most people think about it, it's just that process of getting to the first or maybe the first couple of ribonucleotides. And then we begin, I would say once sigma dissociates, then we transition from initiation to elongation. Okay, keep in mind the bacteria don't know these terms. Okay, these are just ones that we use. Okay, so we finished. All right. So then elongation, as far as we can tell, the rate of elongation is pretty much the same for all of the genes. So once the process starts, it moves along at the same rate through the synthesis process. We won't go into that into any more detail. What we want to look at is this termination process. So how does the system know when it's time to stop synthesizing the RNA, when to let the RNA release it from the RNA polymerase complex, and the RNA polymerase complex has to dissociate from the DNA, and in many cases will cycle around and initiate again. Okay. So these are basically what I just summarized what we'll have to do. There's two major bacterial termination mechanisms. A RHO-dependent termination, which suggests there must be a RHO-independent termination. Okay. RHO is a protein. So RHO is just a protein that's going to facilitate dissociating the RNA from the DNA complex and releasing it to be used for translation or whatever other process it's doing. So this is just a mechanism of terminating the RNA synthesis that doesn't require an extra factor, doesn't require a protein, in this case the RHO protein. So we'll look at this RHO-independent first. The key here is to destabilize the structure. First, slow down the RNA polymerase, and then basically gum up the works to the point where you can destabilize it to allow the dissociation of the RNA. So what you find when you look at the termination sequences, what are the DNA sequences in the gene, that kind of red color, whatever it was when we drew it, what are you going to see in those sequences? Well, for the row independent, that is, it doesn't require some factor, what you're going to find is our old friend, an inverted repeat, followed by a series of about 6 A residues in the DNA sequence. So what this is going to do is they're going to be transcribed as U's, right? And the A-U base pairing is one of the weaker interactions. So if we can first slow down the whole RNA polymerase complex and then destabilize the interaction between the RNA and the DNA, then we have a better chance of getting the RNA released. So what happens is we have these inverted repeats in the terminator sequence. When they get transcribed, they can form this inverted the inverted repeats form this stem loop structure. That starts to slow down and impede the progress of the RNA polymerase, and then that's followed by this series of A's in the DNA, which get transcribed into U's, and because the A-U base pairing is some of the weaker base pairs that are available, it allows this to dissociate, and the RNA can come off, gets released from the DNA template. Okay, you buying that? Sure. Okay. Okay, so here we see, okay, so a little more. So that's basically what's happening here is first you stall the system, and then you have weak interactions, and it allows the RNA to dissociate. Of course, the problem with that is what's going to happen if you start having mutations in that inverted sequence or in that series of A's? Will that affect transcription? Probably would. So there'd be a selective advantage for some sort of an enzyme-based system that could basically terminate any of these transcripts. And so that's really what you see in the row-dependent terminators. All right? So they really have two common features. The whole idea is to try to get the polymerase to pause. What you see is different, and I think, I'll tell you in a minute. What you don't really have is you don't have those inverted repeats. You don't have to have that structural component of the RNA to help get it dissociated because this row protein has a helicase activity. It's going to be able to unwind the RNA from the DNA. And so this is one area where this figure, I think, it's a little bit misleading. I'm not sure why they've put this hairpin loop in here. Because basically, the row protein just moves towards the it interacts with the RNA, it moves towards the 3' end of the transcript, and it has helicase activity. So it can unwind the RNA from the DNA. So you don't really have to have some sequence-specific, you don't have to have some sequences that give you some specific structure to the RNA, and so you have a broader range of RNA sequences. Mutations in that inverted repeat and in that A sequences probably would have less effect on these transcripts. So the main thing is, if you're trying to decide whether you're looking at a row-independent or row-dependent terminator system, look for the inverted repeat in the series of A's. Okay? You look for the inverted repeat in the A's. If you see that, it's probably a row-independent because it's based on being able to form that inverted repeat and have that series of AU interactions which are unstable. If you don't see that, then you're probably looking at a row-dependent termination. Yes?
So, a row-dependent, the row protein is moving faster along the RNA strip than...
Yeah, it actually probably interacts toward as the transcript is getting toward the end. And it's kind of envisioned of actually translocating up along the RNA molecule. But the key thing is it has a helicase activity. So it can easily dissociate the RNA from the DNA. Kind of the difference between trying to build a house with a handsaw versus power tools. Both get the job done, but one's a whole lot more efficient than the other. Okay. All right, let's try. I guess this is more for a breather than anything else. So. Well, we'll go to 1 minute. The more I look at this question, the more I don't like it. Okay. So, 3. All right. So what do I think the answer is? A, but I think you, hang on. Been looking at this. You've got the DNA, you've got the RNA, and you've got the RNA polymerase complex here. It's got to be interacting with all of those entities at some point. So I'll give you credit for A, B, or C. No credit if you put down D. Why not D? Sigma should be long gone by that point. Okay? And then I got to rewrite the question. I'm sorry, what? And actually, this class has been doing better than previous classes of not answering E to a question like this. There always seems to be that 1 or 2. Okay. Uh-oh. All right. How many, this chapter, how many are finding this chapter a little bit challenging? This is what we've been talking about? Oh, you're not going to like this next part. Stay with me. All right. In eukaryotic cells, the process is more complicated. It's always, every one of these molecular processes is always more complicated in eukaryotic cells. So, as we said earlier, in eukaryotic cells, you have the 3 different RNA polymerases. They transcribe different classes of RNA, and they all recognize and interact with different promoters. All right? It's going to be more complicated, and there's going to be more players involved. So try to stay with me here. Okay. First of all, we got this supercoiling, this chromatin. We got this heterochromatin. And so we've already talked about if you're going to have transcription, you have to change the conformation and get the chromatin in a more open euchromatic configuration to give this RNA polymerase complex access to the DNA. So we have all these histones and such, so we have to relax that chromatin first. And so there's a number of, we'll just give you a couple of examples, a number of proteins that are really functioning to change this conformation of the chromatin. We haven't even started to transcribe yet. We haven't even found the RNA polymerase promoter. We're just trying to change the conformation of the chromatin. Okay. There's a group of enzymes called acetyltransferases. The key point here is those histones are basic proteins. They're positively charged at physiological pH. That positive charge interacts with the negative charge on the DNA to help neutralize that negative charge, to reduce that repulsion you're going to get from like charges. And so the chromatin in its tighter conformation is going to have those positive charges. What we'd like to do is kind of relax that a little bit, maybe not have those histones binding so tightly to the DNA. So all the acetyltransferases are going to do is add this acetyl group onto the amine groups that are on the end of these lysines and argines. Take-home message is it takes away the positive charge, puts a no net charge on it, and now when these proteins are acetylated, they are no longer positively charged, and they don't interact with the negative charge on the DNA. And so what you see is in histones, when the chromatin is in this looser configuration, you have more of these acetylated histones for the purpose of neutralizing the positive charge.
You said it takes away the positive charge and puts the neutral charge back.
Well, what they're doing is it's basically the acetyl group's being put on an amine that's out on the end of the R. So what was positively charged now has this acetyl group which has no charge.
And it doesn't.
So the histones no longer have that positive charge. They can't interact to neutralize the negative charge on the DNA. So that now you have more negative charges on the DNA. You're going to tend to get more repulsion from these like charges, and it's going to help the chromatin move in to a looser configuration. But that's only a part of the story. There's a whole other group of proteins called chromatin remodeling proteins. We'll just talk about them as a group. They're really there to the same sort of thing. Sometimes they have to move some of the nucleosomes around, but they're all functioning to transition the chromatin from heterochromatin into the euchromatin. So back when we did those talked about those DNA sensitivity experiments and the globin and in the erythrocytes, and we showed that the hypersensitive sites were changing, it's because the chromatin conformation is changing, and these chromatin remodeling proteins and acetyltransferases are just some of the enzymes and players involved in making this happen. We're not going to go into it in any detail. All right. Let's assume now we can actually get access to the promoters. In general, the organization is going to be similar to what you saw in prokaryotic cells, except there's just a lot more players and a much wider variety in these promoter sequences. We're really going to talk about two types of promoters. Let's say, there's the promoters, and then there's a special class of promoters called enhancers. Okay. So the traditional promoters are just like in E. coli. So they are adjacent to the transcriptional start site and probably extend no more than a few hundred base pairs. So they maybe go out no further than about -200 or so. Of course, to make it complicated, there is an exception. This kind of surprised people when they found it. The promoter for RNA polymerase III is actually downstream in the structural genes. Okay, but that's the exception. And that's about all we're going to say for that. The other important point in these promoter sequences, because I got to make a distinction when I tell you about enhancers, is they have a very precise fixed location. So just like with the bacteria, if you start to move those sequences, you will change where RNA transcription begins. You may block it all completely. If you were to flip the sequence over, it would be non-functional. So it has to have a proper orientation and a proper spacing, just like we saw in the bacteria. So when that whole story, yes. Oh, I'm sorry.
So is the promoter for RNA polymerase III transcribed?
Technically, in that case, yes. It's sort of a funny situation. The only example I know of where it's actually downstream. All the rest of them are upstream and aren't transcribed. So once investigators kind of figured that story out, and I believe these were the first enhancers were found in SV40 as simian virus. So basically a eukaryotic virus that infects monkey cells. They started looking at the structure, and what they found were DNA sequences that could dramatically enhance the rate of transcription. So they were like promoters, except they weren't always located near the transcriptional start site. They could be thousands of bases away. Sometimes they were upstream. As people started finding more of them, they started looking. At first, maybe like, okay, is this something really novel to this particular virus? So they started looking in eukaryotic cells, regular cells, as we think about them. They found these enhancers. There's lots of them. Almost every organism where they've been looked at, they've been found. They were upstream, they were downstream. Some of them were sitting in introns in the middle of the gene. As long as they were within a few thousand base pairs of the gene, they seemed to work. It also didn't seem to matter which way you put them. You could flip them over, kind of move them around a little bit, and they still did their job. So enhancers were really promoters which had a lot of position independence. And so like I said, it can even be upstream, downstream. Some of them are even within introns, within genes. Okay. So you got the promoters, which are the critical sequences that are right adjacent where transcription begins. And then these enhancers, I'll show you a little bit later how that works, that enhance transcription, but they have a lot of position flexibility. The other is there's no sigma factor in eukaryotic cells. So sigma was what gave the specificity to the DNA polymerase and told that holoenzyme where to position itself on the DNA. In eukaryotic cells, the equivalent of the core enzyme structure, can find that promoter on its own. The main thing here are these accessory proteins or transcription factors. So when you see the term factor, you're talking about a protein. These factors are going to bind to elements. That means these are proteins that are going to bind to specific DNA sequences. And it's these transcription factors, or sometimes called transcriptional activators, which are going to be necessary along with the RNA polymerase complex in order to initiate transcription. Okay. Okay, let's try this. Okay, that's not, yeah, maybe it's bad. Okay. We're going to have the general transcription factors. So this is going to be what you're going to find. Let's just call it your run-of-the-mill generic eukaryotic gene that's going to be transcribed by RNA polymerase II. We'll just focus on RNA polymerase II. So this is what's going to be in common with this, and this will be the basal transcription apparatus. So it's going to be a large complex, lots of polypeptides. We're only going to talk about a few of them, but this is going to be in common. It's going to be present to transcribe virtually any gene within the eukaryotic cell. The transcriptional activator proteins are going to be needed in addition. That is, the basal transcription apparatus can find the promoter. It can be locked down on the DNA, ready to go, but essentially has almost no activity until these transcriptional activator proteins bind to their DNA sequences. So that's how the cell is going to be able to regulate when these genes are turned on and turned off by whether or not these transcriptional activator proteins are present. All right. So like I said, we'll deal with RNA polymerase II. So we'll break down, like what you should do, take something complicated, let's break it down into smaller pieces. The promoter is going to have two components. The core promoter is going to be immediately adjacent to transcriptional start site. That's where this basal transcription apparatus is going to bind. The regulatory promoter is going to be where these DNA sequences are that the transcriptional activators bind. And you have to have both to make the system work. Okay, if you look at the core promoter, as I said, it's immediately upstream where transcription starts. It has consensus sequences, very similar to what we saw in bacteria. So this one's been called the Tada box. It's between about -25 and -35. The position's a little different, but you'll see the functionality is the same. There's going to be a protein that's part of that basal transcription unit that's going to be looking for that sequence, that TAT-AAA sequence, to tell it where to lock down and position. So I kind of think about it, I mean, if you've ever done any kind of, you guys build furniture in that, right? No, you go to IKEA, forgot. Okay. But basically, use these little pegs and dowels and such, and that's to get everything to line up. The same thing's happening here. Here's the little hole. The peg is going to be this protein that's on the basal transcription apparatus. It's going to position it correctly at this -25 to -35. Just like we saw with the bacteria. If you start mutating those sequences, you dramatically affect the efficiency of transcription. If you start moving them around, you change where transcription begins. So it's a molecular marker also that tells the system where to start. Okay, so here's your sort of basic eukaryotic core promoter. Transcription starts here. This is just the one as an example. This is just for RNA polymerase 3. It's actually downstream. But if we're just going to focus on RNA polymerase 2, so here's transcriptional starts here, and here's the Tada box, and this is the recognition sequence for a protein called TF2B. I'll tell you about that in a minute. Okay, and then the regulatory promoter is going to be even further upstream. That regulatory promoter is where these transcriptional activator proteins are going to interact. The key to this is, in eukaryotic cells, you have to have more ways to regulate gene expression than simply making 2 or 3 different sigma factors. So what you have here is a whole array of transcriptional activator proteins, a corresponding set of the DNA elements that these proteins are going to bind to, and how in the combination of how those proteins combine can let you control when and where you're going to express these genes. All right? But these transcriptional activators have to talk to the RNA polymerase complex. So there's a protein on there called the mediator, which is just really the link. That mediator protein is going to interact with these transcriptional activators. So when that whole chain is present, it activates transcription. All right, I'll show it to you in a diagram in a few minutes. All right. So if we took the diagram I had before, now we look a little further upstream, here are the regulatory promoters. So these are basically these enhancer sequences. Some of them are enhancer sequences. These are just different sequences which these transcriptional activator proteins can bind to. But because you have a significant number of them, you can put them together in lots of different combinations. It gives you lots of different ways to regulate gene expression. Okay, let's try some real simple math. Let's do this real simple. Each one has really got 2 states. Either the protein binds or the protein doesn't bind, right? So let's say you have 8 of these transcription factors. Well, 2 to the 8th is what? From all the computer geeks. Isn't that 256?
Yeah,
256. So if you have 8 transcription factors, 8 binding sites, you got 2 states for each one, that gives you 256 different combinations. We have a little over 200 different types of cells. So with as few as 8 transcription factors and 8 binding sites, you can have a unique combination for every cell type in your body. So it's through this kind of interaction in putting them together in different arrays that lets you control gene expression. And you'll see they're not each unique to a different gene. Some are, and some aren't. Okay? But the basic idea is these transcription factors are going to bind these DNA sequences, and that's what's going to activate gene expression. Okay. Just to make a comment about RNA polymerase, we already talked about RNA polymerase 3. We'll just say RNA polymerase 1. The system's very similar, but let's really focus our attention for RNA polymerase 2. Okay. All right. Here's where it gets a little wordy, and it looks like an alphabet soup down here. Let me see if I can kind of break this down for you. All right. So we're going to have this basal transcription apparatus. This is the assembly that you're going to find associated with the core promoter on your basic, generic, run-of-the-mill eukaryotic gene, RNA polymerase 2. So we're going to have the transcription factors, RNA polymerase 2, and this mediator protein. So here's these some of the transcription factors. TF2A, TF2B, on and on. Well, the first thing is TF2 just tells you it's a transcription factor for RNA polymerase 2. So they're all the same that way. So it's really just the letter that makes a difference. So we'll just look at a couple of them. TF2D Now you got to follow with me here because it's going to get confusing in a minute. TFT do, see, I told you. All right. TF2D, that protein, that's what binds to the Tada box. All right. So it's looking in the promoter. It's the key protein. That's the peg that's looking for the hole in the DNA to lock it into place. All right. Well, TF2D isn't just a single polypeptide. It's actually a complex. It's a multimeric complex that is 9 polypeptides. One of those peptides is the Tada box binding protein. So that's really the peg. So TF2D is at least 9 different polypeptides. One of them is TBP, the Tada box binding protein. Now, going all the way back to whatever it was when we talked about the DNA chapter it was on DNA structure, I said proteins can read the DNA sequences by interacting in the grooves of the DNA. Well, the Tada box binding protein binds to the minor groove of the DNA, finds its DNA sequence, and I think I also showed you a diagram, actually begins to bend. The binding of that protein complex is going to begin to bend the DNA. What's going to happen when you begin to bend the DNA? You're going to make it easier to break the hydrogen bonds and open the helix, which is the first step in transcription. So the binding of this complex is starting to weaken and make it easier to unwind that helix. All right, so that's what it's trying to show you here. And so here's that Tada box binding protein. Okay. Okay. Now there's Tada box binding protein associated factors. All right. Those are all the other polypeptides that come together. That gives you TF2D. So all the other proteins except for the Tada box binding protein are these associated factors. All of those all together, at least 9 different polypeptides just to create TF2D. All right. The holoenzyme is going to be the RNA polymerase, the enzyme, actual enzyme activity, this mediator protein, and all these other transcription factors. Everything is thought to pre-assemble before the TF2D transcription factor. So you're assembling, before it even interacts with the DNA, you're assembling this whole basal transcription apparatus except for TF2D. Some of these other ones, just to give you a couple of them, TF2A is just there to help stabilize the interaction because this is so critical to help stabilize the interaction between the Tada box binding protein and the DNA. TF2B is there to help the complex recognize the correct start site, the +1 where transcription will begin. And the one that's kind of easy to remember, TF2H, is the helicase activity that will begin to unwind the DNA. Okay, so you can see this from the back, right? Yeah, right, okay. Okay, so here is the transcriptional start site, the core promoter, the Tada box sequence right there about -25, -35, and the regulatory promoter. This is where all the DNA binding elements are for the transcription factors. So we're going to have TF2D, and right one of the proteins there, right there, is the Tada box binding protein. So it's going to bind to that sequence. Here's all of the other members of this basal transcription apparatus with some of the other factors that we talked about. Here's that mediator protein that's going to help this whole complex talk to and interact with the transcription factors. All right. And this is why the enhancers work even though they are upstream or downstream or someplace else, because unlike the diagrams that we put in the book where we draw DNA as nice rigid lines, It's actually a flexible molecule. The whole idea is that here's this mediator protein. If the transcriptional activator bind is present and it binds to its DNA element, then that protein can interact with the mediator. So these transcriptional factors tend to have two active domains. One is a DNA binding domain, and the other is a protein binding domain. They can interact directly with the mediator, or they may need another mediator. The co-activator is just another linker protein. But in one way or another, you have a protein to protein to protein interaction, so that this basal transcription apparatus at the molecular level knows the transcriptional activator proteins are there. Okay. And that's that part. That one may take a couple of your beverages of choice to work yourself through. Okay. Elongation, as I said, we'll omit the discussion on that. It's basically, as far as we can tell, just in eukaryotic cells also, initiation is the rate limiting step. Actually going from that closed to the open transition, the open initiator, where the helicase can begin to unwind the DNA, that's the rate limiting step. Looks like elongation, the synthesis of the RNA, occurs at about the same rate for all of the genes. So let's talk a little bit on the termination side of it. So it may be a little more straightforward in eukaryotic cells than it was in bacteria. In eukaryotic cells, we really don't have a defined, most genes don't really have a defined termination sequence. What happens is the transcription goes past the point where the RNA needs to be made, and I can probably, and then there's going to be an enzyme that's going to come along and clip it at just the right place. So since it's just trimming off the 3' end, it doesn't really matter where it actually terminates as far as transcription goes. So maybe it's a little clearer here. So we have the RNA polymerase, we have transcription. What you have is a recognition sequence where this clipping enzyme, which I always kind of like the name, the RHO-1 endonuclease, all right, is really going to attach to the RNA. Well, it'd be the pre-mRNA. We're talking about RNA polymerase 2. Finds the sequence that tells it now a certain number of bases downstream, just clip the RNA. So termination, and so what you look at, if you were to look at if you looked at the same gene, multiple rounds of transcription, look at where termination occurs, it would actually end at different points. Because it doesn't really matter because they all get trimmed off at the point that's required. And then it clips, well actually, so there's a clipping enzyme, there's a DNA recognition sequence that clips the RNA, and what this RHO-1 endonuclease is really doing is just removing these fragments of leftover RNA. So it just works its way up the RNA, degrades it, all down to ribonucleotides. So the important point here is, unlike the bacterial system, there really aren't defined termination signals, but rather it just sort of terminates. There's a sort of loss of processivity as long as it gets past the point where the clipping enzyme comes in, so all the mRNAs are clipped at the right place. Okay. Let's try this. All right. Mm-hmm. I'll go to 1 minute on this one. Okay, what's the answer? Say it out loud because I forgot to put the slide up with the answer. I go with C.
How's it?
They don't modify the chromatin. All these it's a good point. Modifying the chromatin was there to, on a much more global scale, to just get things released. The transcriptional activators are working very precisely to initiate transcription of specific genes. So they're going to end up interacting with the RNA polymerase II through the mediator protein. They can bind either those regulatory promoters, which are fixed in position upstream of the core promoter, or these enhancer sequences. They're doing the same thing. And their job is to increase the rate of transcription. Yes? Well, you don't have an RNA polymerase II in eukaryotes. In prokaryotes? It's a fine point. In eukaryotes, transcriptional activator proteins are not part of the holoenzyme. They're interacting with it. So again, it's these limitations of writing these questions. So they end up forming an interaction with the RNA polymerase II complex via this mediator protein.
But they're actually part of the holoenzyme?
Not initially. So yeah, A is probably wrong. God, I hate multiple choice questions. Tell me what you know. Well, in this case, they do all of the fun. In this case, I'd have to give credit for A and C. Because A is, it's really an indirect interaction. It's just trying to function in a world where you have to describe a complex process in one sentence. Okay.